Thanks for giving me the opportunity to present my PhD work here. So the production of crops, they secure human food supply, as we all know, but climate change is bringing new challenges. Plants and corresponding environmental data are super important to uncover crops' responses to changing environments. And this can be done with essentially two tools, phenotyping and modeling. So that's why I also have highlighted them in the title of my thesis, which was exploring plant responses to changing environments, integrating phenotyping and modeling across scales. So yes, phenotyping. I mean, everyone in PhenoRob probably know what phenotyping is, and it's emerging from the term phenotype, which was first used by Wilhelm Jørgensen. That was a Danish researcher some time ago, who describes a wide range of descriptive characteristics in plants, microbes, and fungi, and of course also animals. In the 1960s of the last century, the term phenotyping was then introduced from the term phenotype, which refers to a quantitative trait analysis of characteristics of a certain organ. So basically, plant phenotyping is the comprehensive detection, recording, and analysis of observable plant traits, which are related to growth, development, and, for example, also stress responses. Modeling is basically something a bit different because if you want to observe complex structures, it's sometimes complicated to get this done experimentally. And plants are complex systems. So we use models to simplify and represent complex systems, allowing us to study, analyze, predict, and also optimize various aspects of the system in a kind of controlled and efficient manner. So the domain which we are using in plant science, the kind of models, is, for example, functional structural plant models. So short FSPM, and this is a kind of computational plant modeling. The structure is simulated based on different roots, and the physiology of the plants, so functions basically, process-based plant functions, they are integrated into the structure, which then enables the representation of, as I said, the physiology. So there are different physiological aspects of the plants that can be simulated, for example, carbon allocation related to photosynthesis processes, the water flux inside the plant, nutrient uptake of the root system and the distribution within the plant, and so on. The combinative part of phenotyping and modeling allows us then to access the plant responses to various conditions on various scales. So various conditions can be, for example, climate change, reduced availability of water, reduced availability of certain nutrients, such as ending nutrients as phosphorus, for example, which is mined from rock, and therefore we have only limited resources, or also nutrients that get too expensive, for example, certain nitrogen fertilizers, and so on. But as I said, it's done on various scales. So I did it on various scales. So what scales am I talking about? If it comes to during experiments, there are essentially three scales in plant science. Of course, there's the field scale. That's where we do agriculture, and that's where we grow the plants for human nutrition in the end. There's the greenhouse, which is the scale where we experimentally look a bit more detailed on the plants. So that's the scale where we look at processes, at plant interactions on a small scale. That's the scale where we look at precise disease reactions and so on. And there's the modeling scale, also often referred to the in silico scale. And this in silico approach gives us a bit more detailed information about the processes, because we can simply switch on and switch off different functions and different structures of the plant, and therefore look a bit more distinguished at different processes that we want to observe. During my PhD, as I said, I covered all of these scales. So there's another scale, which is the organizational scale, if you look at the plant itself. And it ranges from molecules, genome, there's the plant physiology, which is probably the most known scale in plant science. There's the organ scale, if you look at different organs such as leaves, root types, so primary roots, similar roots, for example, the crown roots, or the stem itself, the hypocotyl. There's so many organs that you can look on. And there's the ecosystem scale. So the ecosystem scale is basically the interaction of different plants with the environment and each other. In the group where I did my PhD, we mostly modeled on organ scale, but we can also upscale a bit on multi-plant scale, which is then basically a field, which gives us certain insights into the ecosystem. And because we are using functional structural plant models, we are also able to incorporate physiological aspects by the functional modeling part. So this is the basic overview about the domain of science I was working on during my PhD. And now I would like to share the objective of my PhD. So the idea behind my PhD was to improve the structural and functional quantification of plants with a special focus on their root systems. And this was essentially done with a very important plant for human nutrition, a very important plant for agriculture itself, and a lot of different other aspects which we have in plant production, for example, fiber, fuel, and all this stuff, livestock, nutrition, all this. So this is maize, which I consider the backbone of agriculture somehow, since it's the most harvested plant in the moment. And I centered my PhD thesis around 4 topics. The first of all is, in order to understand root development over time, we require field data. And it's important to understand how the plants, and the roots especially, develop under real conditions, real environmental conditions in the field. And it's quite complicated to do this because roots are hidden by soil and they are developing inside of the soil. So if you want to observe them, you either disturb them or you try to observe them in a kind of non-invasive or minimal invasive manner. And this is quite complicated. So there are several techniques to do so, but I will explain a bit more about this later. So the first idea was to develop and validate an automated image analysis method, so a kind of non-invasive approach to improve the efficiency in root phenotyping itself, because it's, as I said, quite complicated to get non-invasive root data over time. Then the second thing was that I wanted to provide a comprehensive subsoil data set for very scientific applications. So basically, as I said, you need plant data and environmental data to understand how plants are responding to changing environment. So for example, climate change. And yeah, a comprehensive data set or comprehensive data on this over time, observing the plant development into certain environmental condition over maybe a decade even, is very important to understand the dynamics which drive these changes. And since maize is very sensitive to the absence of certain nutrients, and of course also to water scarcities, I wanted to understand how maize responded to phosphorus deficiency. Therefore, I wanted to investigate the maize responses to phosphorus deficiency and quantify the changes in the architecture of maize and how this is related to the water uptake capacity. So basically somehow cover 2 of the most sensitive problems of maize, which is the deficiency of phosphorus and the absence of enough water. And in the end, the last point was to transfer my approaches, so basically the phenotyping and the modeling parts, to study maize responses under environmental conditions in the broader context. So yeah, let's start with the first part, which was taking place on a kind of experimental scale and was looking basically at the organ and the ecosystem itself. Therefore, I would like to briefly introduce the Seelhausen field mini-rhizotomes. So some of you who were working in PhenoRob may have heard of the mini-rhizotome facilities of Seelhausen. These are root observatories where you can basically observe roots in the soil developing. And these facilities are situated within the Tirino Eifel Lower Rhine Observatory near Seelhausen, Germany. That's where the name, of course, comes from. And they are built to compare different agronomic treatments and are therefore divided in several plots. Within the individual plot, so here on the left you can see one individual plot, there are transplant tubes installed in the soil at depth between 10 centimeters and 120 centimeters and a length of 7 meters. So beside other sensing methods, which I will explain a bit more in detail later, there are camera systems guided inside of these tubes, which can capture basically the root development dynamics by taking images. However, as you can imagine, if you do images over time in all these tubes, so we have per plot, we have 18 tubes, we have 3 plots per facility, and so on, you get a lot of different, images. So the data set is consisting for each measurement period of 10, maybe even 100,000 images. And we had kind of an image analysis problem there, because it was very time consuming to analyze these images, because it was actually done manually, because these images are very heterogeneous and it was complicated to extract the roots from the background. And since it was done manually in the past, it was done by hand tracing, which is also subjective. So there were many annotators, many students and PhDs with a different view on how a root should look like, if a root is alive or if it's already dead. And of course, this gave a certain bias. And there was only limited information from this manual analysis, because it only gave us the root length basically in centimeters as the only trait from the image. And the idea was to find a solution which was based on the automation of the process. And the key requirements for this process should be that it is fast, objective, and has a high informative value. So basically reducing processing annotation time, getting rid of the factor human as good as possible, and increase the number of root traits that could be extracted from the images. The solution I found in a neural network-based root segmentation, which was then implemented on our house cluster and also at the UD supercomputer for upscaling. And there we extracted and segmented the roots, and then later executed automated feature extraction, which gave us a lot of different root traits then from the images. So this is the pipeline. So basically it starts with getting the minute rhizotone images, measuring in the field, then extracting a small image sample, do some neural network training with it, and then do the automatic segmentation based on the models that came from the network training. Then there was a conversion required of these images to binary, basically. And then this binary data set could be put in a batch analysis system with program called RhizoVision Explorer, which resulted then in a handful of different features. And they developed over time, as you could see here. So these are the rhizotubes in the soil, and the red dots, I hope you can see them in the stream, the root length density at each observation point that we had measured at different time points. So we did this pipeline, we let it run for almost a decade of data, and then looked at a small amount of validation data. And from these validation data, we could see that basically we had only less than 2% of the time we required for manual annotation that it takes us to process the data with a new automated method. We then compared the correlation to the manual analysis system that we had before, and it was an overall very high correlation of 0.9. And as I said, the next good thing is that this automated analysis method is now providing additional root traits. And maybe one of the main outstanding points of the study was that we have enough labeled data, because the bottleneck for neural network training, to avoid the buzzword AI training, is to have enough labeled training data and ground truth data for model validation and for model training. I'm quite happy that this methodology is now kind of the state-of-art approach for in situ root image analysis in a lot of different studies and also research facilities, including a lot of field trial sites, also in international universities such as, for example, Kentucky, Negev, I think Bonn you also use this, and Copenhagen, as well as our institute, of course. Yes, now I would like to stay at this scale, so at the field scale and the organ and ecosystem scale, and would like to broaden up in the data structure a bit. So in order to deepen the understanding of root system development over time, data from roots and, as I already teased at the beginning of the presentation, from the environment are required. So at the Seelhaus mini rhizotome facilities, we gather data from different sensors, not only from the root observation cameras. I already briefly introduced the Seelhaus mini rhizotomes. However, there's more than these cameras. There are, first of all, there are 2 facilities located in different soils. So there's one in a very loamy soil and one in a very stony soil, which is barely usable for agricultural production in these areas, but still there are a lot of, oversea there are a lot of soils that look like this. And these 2 facilities are built in the same way, but they are located in the same climate because they're just a couple of 100 meters away from each other. The only thing that differ is the soil type. And we observe, of course, the root development, but there are also soil moisture sensors, which give us information about soil moisture. So there are TDR sensors installed at permanent positions in the soil to give us static information about the development of soil moisture over time. There are TDRs, matrix potential sensors as well for logging also, of course, the soil metric potential, but the temperature. There are tensiometers, which are also giving soil water content and soil metric potential at a different range. And there are, of course, the rhizotubes, as I said, which we take for image analysis. So this is how it looks like when I'm taking the images in the rhizotome facilities. And for GPR, which is ground penetrating radar measurements, which is done also within the institute I'm working at in Jülich. So these data have been analyzed in the past by a lot of different PhD, postdocs, professors, and used for different studies, but they have never been brought together as one big data set. So one of the key achievements during my PhD was basically that together with my colleague Lena Lern, I created one of the most comprehensive and extensive multi-year plant soil atmosphere data set based on these experimental data from the mini rhizotome facility. So it was basically a lot of labeling, standardizing, and looking up for data, reanalysis of data. And we did all this together for the data from 2015 until 2023 now. So it's almost a decade of data that we have. And we published the data then in 2022 with data containing experimental data from 2015 and 2021. This is a running database which is always updated, and it contains different experiments such as drought stress experiments, sowing density, crop mixture experiments with deep and shallow rooting cultivars, for example, and recently also nitrogen experiments. All this was published open access, and the nice thing was that we did a collaboration with the University of Bonn and PhenoRob, of course, and we could combine our subsoil data of roots, GPR measurements, and soil moisture with measurements from the same experiments that were taken at the University of Bonn, or of colleagues of the University of Bonn at the same facility, and we could combine them to have like above and below ground data set in the end. Okay, so this was basically the experimental field scale that I presented so far. Now we are switching a bit the experimental scales from the field to the greenhouse and to the computer, or inside of the computer, to do some in silico plant analysis. And also we dive a bit deeper into the plants by not looking at ecosystem scale anymore, but looking at the organ scale, so the structural organization of plants and the physiological aspects. And it's still about maize, since it was the model crop I used for a lot of things during my PhD work. And as I already said in the beginning, maize is quite sensitive to phosphorus deficiency. And this is because there are different processes going on in the plant, which are not completely all known and well understood. And the problem is that maize is sensitive to phosphorus deficiency in at least 30% of the cultivated land area where maize is grown. And phosphorus availability introduces certain changes in the shoot and the root system architecture, which then lead to reduced yield in the end, for example, but also to bad development and generally bad agricultural conditions, you could say. And under field conditions, most of these plant responses are measured in rather coarse metrics. So, for example, they just look at the yield or at the leaf area or maybe at the fresh biomass, dry biomass, whatever. And we do not obtain real direct response functions for the development of certain organs or certain physiological functions of the plant. And it's although we have a variety of different plant responses that were already reported, such as reduced growth of the shoot, for example, it remains a bit uncertain, or remains so far a bit uncertain, which parameters have the other driving forces behind these changes or these coarse changes in the structure of the plant. And it's very complex to transfer architectural changes to the functionality of the plant itself. So it's even more complicated if you already have the changes of different organs due to phosphorus deficiency, to transfer what that means for example, water uptake of the root system. So the solution for this problem was to use a combination of experimental data and modeling, so getting the structural data from experimental greenhouse trials, and parameterize a functional structural plant model for the whole plant, and then see, and this is the first main objective of this certain study, which structural parameters of maize organ show the strongest response to soil phosphorus availability. So we basically wanted to have the driving forces behind the changes. And also we would like to understand how the root system conductance, which is a very important proxy for water uptake capacity of the root system, and therefore, of course, maintaining function under drought stress in maize. And we want to know how this adapts in different soil phosphorus availabilities. And for this reason, we set up a greenhouse experiment, which was done in rhizoboxes. We had a size from approximately 80 to 32 centimeters, and we took phosphorus deficient field soil from the Dickhoff experiment and added different levels of fertilization to these phosphorus depleted soil. So we had like phosphorus depleted soil, which was the original soil, an additional amount of phosphorus, which was then mild, a severe phosphorus deficiency, a moderate phosphorus deficiency, and a very mild deficiency, and of course a fully sufficiency. And we then analyzed the images that came from this rhizobox experiment by the image analysis pipeline I introduced before, and performed an organ tracing on these segments then, which gave us parameters that we could then analyze and understand which structural responses we had. And also we would like to parameterize the functional structure plant model with these parameters. Yeah, so to parameterize and use a maize functional structure plant model from experimental data is quite complex, because there are several parameters that need to be considered. I will not go into detail here, but all these parameters that are explained in this overview were analyzed from the images for each organ. So for each root type and for the leaves, for the stem, and so on. Additionally, there was some global information about general shapes of the leaves or amount of different root types that were also obtained. From all these parameters, in the end, there were only 3 parameters of the whole plant, which were significant response parameters to the deficiency of phosphorus. And in the root system, it was basically the axial root radii, which was increasing with increasing amount of phosphorus in the soil, and the ground root elongation, which was also increasing with increasing amount of phosphorus, but it stopped at some point, so there was plateau at some point. And the shoot, which was also responding to the phosphorus deficiency, responded with a decreasing leaf elongation with decreasing phosphorus availability, which is not very much surprising, because if you think of phosphorus deficiency, you think of smaller leaves and smaller shoots, some of these rigid appearance. We took all these parameters for model parameterization and parameterized a functional structural plant model based on C-Plantbox for the different phosphorus deficiencies. So we came up with 4 different model types, which were all stochastic. So they had a variation, which should somehow cover the natural variation we have in the maize genotypes. And we did this for 4 weeks of growth, because this was also the experimental period that we could observe in detail. And in this image from the model runs, so these are the visualization of the different model runs of low phosphorus or almost no phosphorus to phosphorus sufficiency, you can already see that there are different, like visual differences in the appearance of the plant. And we wanted to know how the water uptake capacity, so namely the root system conductance of the maize plant, was changing. So we added also some hydraulic parameters to this simulation. So we combined the architectural models with anatomical root data to parameterize the seed plant box model to enable, beside the dynamic growth, also dynamic root system conductance calculations under the different phosphorus supply regimes, of course. So we had axial conductance values and radial conductivity values from another study of Heymans et al. From 2021 from the University of Louvain, who did these for the same mice genotype for the same time frame. And these axial hydraulic conductance values relate basically to the efficiency of water transport along the length of the root, main axis. And the radial hydraulic conductivity of a root segment is the measure of a root's ability to take up water from the soil into its vascular system. So it basically represents the water uptake potential by a root segment for a specific root surface and water potential gradient between the xylem and the outer surface of the root. And these data together can be used to calculate the KRS based on the model simulations. And these were the results. So KRS is short for root system conductance. And we see that we have differences between no P, which is the blue line here, and sufficient phosphorus amount in the plants. And we see, interestingly, we have a 40% difference here, but we see that there's no significant difference between any kind of deficiency. So between mild, very mild phosphorus deficiency and almost no phosphorus in the soil, there's no significant difference between these treatments, while there's significant difference between all kind of deficiencies and the sufficient phosphorus supply. And it's interesting because you could say that there might be a biological implication for this. So the biological implication could be that all plants under soil pH limitations with a lower root system conductance decrease transpiration later than plants with high KRS, since they have a lower water use and soil water could not be depleted that quickly, for example, which would be beneficial under certain conditions. For example, if you have like a temporal drought and later sporadic rainfall, so the water supply in the soil is kind of restocking, and then the plant would survive a longer period of drought, for example, with these potential benefit there. And already during the domestication process of maize, it developed root systems with lower root system conductance in regions that are following these climatic patterns, so having like intermediate droughts over a certain time period, and then a restocking of the soil water content, for example, in southwest of the US. And this adaptation could be a potential adaptive advantage under these sporadic dry spells. We also had to validate this model data. So we compared our range of model results with some literature values. We saw, luckily, that we could we stay in the same range, because it's quite complicated to do this experimentally, because you almost can't experimentally compare these data or measure these data, because you have to look at the whole root system distinguishedly, each root type, each little root, and this is experimentally almost impossible. So we were quite happy that this was done, or this was firstly now possible to do in silico. As last point of my today's presentation, I would like to show you a small transfer of work from the PhD project to a broader context within PhenoRob. So it's a nice example of a PhenoRob collaboration, because we took this model and phenotyping and modeling pipeline, which I just introduced, to get root system conductance values and structural information of the plants in order to check whether the similar root numbers of maize was in fact the driving trait for adaptation of maize during its domestication process. So the theory was that during the domestication process of maize starting in the lowlands of Mexico, distributing all over the world, there was a driving trait which was the similar root amount, or the count of similar roots. And yeah, the idea was that this was done to kind of to make maize drought resilience in certain regions where there is less water availability. So the idea was that the drier it is at a certain condition, there are fewer similar roots in this condition expressed. And yeah, we could apply these phenotyping to model pipeline that are just introduced to have like a certain amount of images, like a few hundred images of maize genotypes. We phenotyped them, extracted model parameters from them, and did the simulation with cPlantBox to estimate the root system conductance for the different genotypes. And first of all, we could see, and that was very interesting, that we have a similar root, increasing similar root number, and it comes with a decreasing primary root length and a decreasing lateral density. So this was an architectural relationship that was uncovered until then. And secondly, even most interesting, we had a lower root system conductance in cultivars with lower similar root number, which kind of indicates a more conservative water spending strategy, which is, as I just explained, favorable in dry conditions. So this gives some genetic advancement, because it is quite cool to know these features, because the underlying gene was also identified in the study to breed, for example, drought resilience or climate resilience maize cultivars in the future. So I really like this study because it somehow combined all the scales I had during my work as a PhD, or I worked on during my PhD thesis, from field over greenhouse and modeling, to look at genome and physiology at organ and ecosystem scale. And that's the information, or that's the last thing I wanted to say before I conclude my presentation, by just showing you the PhD highlights of the last 3 years, which were, of course, not only powered by coffee and curiosity, but also by very good and greatly supporting supervisors and a lot of very nice colleagues who helped me during this time and always supported me. A lot of collaborations within PhenoRob, which all made this possible. So I would like to thank you for your attention.